In this class, today we will discuss about the female reproductive organ, that is the gynoecium. Gynoecium is of two types. If it consists of one carpel, then it is monocarpellary. If it consists of more than one carpel, then it is multicarpellary. In multicarpellary type of gynoecium, if the carpels are fused, then it is known as syncarpels, and like in mustard. Or if the carpels are not fused free, then it is called as apocarpels, like we find in lotus or in rose flower. Now coming to the structure of a carpel, it consists of three parts: the stigma, the style, and the ovary. The swollen part, which is sticky in nature, is called the stigma. This is the stigma. It is a platform for the mature pollen grains to fall. The stigma has some chemotrophic substances which stimulates the pollen grains to germinate the pollen tube. This cylinder tube-like structure is known as the style, and this swollen part is known as the ovary. The ovary has got a receptacle or the thalamus. Inside the ovary, ovules are present. Ovules are round small structures. They are present on the placenta. Placenta is a tissue. This is in the ovary. The ovules are present on a tissue known as placenta. The arrangement of ovules on the placenta is known as placentation, which you have already studied in 11th standard. The ovules are actually the placenta, not the ovules. The placenta is placed in the ovarian cavity or the locules. Now the ovules have got a stalk-like structure which attaches itself with the placenta. A stalk-like structure is present which is known as funicle. This is known as funicle. The stalk with which the stalk by which the ovule is attached to the placenta is known as funicle. So this is the funicle here. This is the funicle. The region in which the ovule is attached to the funicle is known as hilum. Now this is the micropylar end. This is micropyle. Micropyle is the place which is not jointed. See, there are two integuments, the outer integument and the inner integument. They are the protective layers. This is the outer integument. And this is the inner integument. Angiospermic ovules have got two integuments, so they are bi-integument. Inside this, the upper region, this upper basal region is the chalaza which is just opposite to the micropyle end. The chalaza actually is the basal part of the ovule. Now inside this, there are parenchymatous cells, a dense parenchymatous cells which are known as nucellus. This is known as nucellus. Now this is the structure of anatropous ovule. Anatropous ovule is always upside down, and majority of the angiosperms have got anatropous ovule. Now students, before we proceed further, we must know what is an anatropous ovule. The body of the ovule is inverted, as a result of which the micropyle comes near to the hilum and faces the placenta or the base of the funicle. The micropyle and the chalaza are in the same vertical line. It is the most common type of ovule found among many angiosperms. Based on the shape and orientation of micropyle and the chalaza in relation to the funicle, angiospermic ovules are of many types. For example, the orthotropous ovule, amphitrophous ovule, hemitrophous ovule, and camelotrophous ovule. Now students, we come to megasporogenesis and the development of the female gametophyte. The nucellus cells develop into megaspore mother cell. This is megaspore mother cell. Of course, this is the chalaza, chalazal end, and this is the micropylar end. This is the micropyle. The nucellus, the megaspore mother cell, will now divide, undergo meiosis. It undergoes first meiotic division, that is M1, and forms a dyad megaspore. It will again undergo second meiotic division and form a tetrad. Now all these tetrad megaspores, that is the megaspore, 4 megaspores are formed. These are haploid in nature. So you can say 4 tetrad linear megaspore. They are linear in structure. Linear megaspore is formed. Now this is the micropyle end and this is the chalazal end. The 3 megaspores formed, which are haploid in nature, they will degenerate and only 1 megaspore will survive. This is in the micropylar end, it will survive. So this is the first cell of the female gametophyte. The female gametophyte develops from 1 megaspore, so it is known as monospory. The surviving megaspore will now function as functional megaspore. This will now be called as functional megaspore. The functional megaspore enlarges in size and the nucleus of the functional megaspore will divide mitotically. This is a free nuclear division where the cytoplasm does not divide. Cytoplasm divides later on and forms the cell wall. So this is a free nuclear division in which only the nucleus is dividing. It will undergo first mitotic division. This is mitotic division. Where a two nucleate stage condition is formed. This is a two nucleate stage. Then a second mitotic division is followed. Where four nucleate stage is made. It again undergoes the third mitotic division where 8 cells are formed. This is a 8 nucleate stage. After the formation of this 8 nucleate stage, cell formation, cell wall formation starts. Now, a female gametophyte, angiospermic gametophyte, has got 8 nuclei. After the formation of this 8 nucleate stage, cell formation starts. This is the chalazal end. Chalaza is there. And this is the micropyle or the micropylar end. The 3 cells at the chalaza end are formed, which are known as antipodal cells. This is antipodal cell. The 2 cells at the micropylar end forms the synergids. And one egg cell is formed. This whole thing, that is the synergid and the egg cell, together form the egg apparatus. Together this forms the egg apparatus. Now one nuclei from the chalaza end and one nuclei from the micropylar end will form 2 polar nuclei. Which forms the large central cell. So a mature angiospermic gametophyte consists of 8 nuclei and 7 cells. It is 8 nuclei and 7 cell structure. 1, 2, 3, 4, 5, 6, 7 cell structure. 3 antipodals are there, 2 synergids, 1 egg cell and 1 large central cell. This is the central cell. And 8 nuclei. Here you see antipodals 1, 2, 3, 4, 5, 6, 7 and 8. 8 nuclei are there. So students, a typical angiospermic embryo sac which consists of 8 nuclei and 7 cells, is known as polygonum type of embryo sac. Students, we have finished microsporogenesis and megasporogenesis. Now the mature pollen grains have to be taken to the stigma for pollination. So in the next topic, we will discuss about pollination.